Hello and welcome to this teaching idea video on how to use a large model within A-level biology to teach the production and release of insulin by pancreatic cells. Right, some of the key features before setting this up is to make sure that you have all the parts produced. Make sure that you've got plenty of phospholipids made and plenty of protein channels made and such forth. Make sure that you're spreading it out over a few tables so the students can gather around and see what's going on. In order to help them as well, make sure that you're using lots of different tiddlywinks and stuff to represent the different ions and the insulin so that it can be moved around easily. Another key point is to make sure that you have lots and lots of different stage cards made to help the students get it clear in the head and break up the process into lots of smaller stages. The following short video will outline how this sort of model can be used.
So, just a few things to remember when going and building a model that you can use with students to help represent how insulin is secreted by pancreatic cells. Right, first thing to remember, have lots of little pieces, right? So phospholipids, different mitochondria, stuff like that. You've got to remember that at some point you're going to need to pack all of this away somewhere. Lots of small pieces will go and help pack it away as they won't take up very much room. Right, next thing to do is have lots of these small stage cards that will go and it'll help prompt you as you go in and explaining through it. After you've gone and you've finished the activities, the students can then go and use the stage cards to produce their own diagrams. Right? So, we start off, go talk about the fact that the membrane has a resting potential of minus 70 millivolts. Right? Use this opportunity to help or get students or question students about how this is similar to the membrane of nerve cells. Right? Then you can talk about transporters. If you're making a transporter, get the shape of glucose molecule, make the receptor sites on that glucose molecule so it fits in. Right? It's just to help students just almost get an image clear in their head about what's going on. Make sure that you've got lots and lots of different, coloured counters or tiddlywinks available that you can use to represent the different ions moving in and out. Because you've got lots of different bits, you can go and move them as you go in and you're explaining. Right? Make sure that when you're doing it, you go in and you're using lots of the key terms and key terminology. At each stage as well, it's quite handy if you go almost stop halfway through or stop at different stages and just ask students questions just to get them thinking about it. So, for instance, you can go and get them to talk about why things are moving down a concentration gradient. What do we call it when a chemical goes and exits the cell? Right? What do we call it technically when something's entering the cell? Stuff like that.
So, now that we've had a look at how to run it, I'm just going to go and show you very, very quickly how it can be used and some of the things that we need to look at when you're using it with students.
So, first thing that happens, before a cell inside of the pancreas can release insulin, a nerve impulse or something needs to happen to it that causes a change. So, normal blood glucose concentration levels, potassium ion channels, right, in the plasma membrane of the beta cells are open and potassium ions can diffuse in and out of the cell. Right, so they can go and they can move in and out of the cell. So the inside of the cell is or has a resting potential of minus 70 millivolts in respect to the outside. So in that respect, it's very, very similar to a nerve cell. When blood glucose rises, glucose enters the cell by a glucose transporter, right? So it'll bind onto it and it will get transported in. Right? The glucose is then metabolized by the mitochondria, resulting in the production of ATP. Right, so a number of, we're not going to talk about it now, but remember a number of different processes will happen that causes the metabolization of that glucose before it produces ATP. So the ATP binds to the potassium ion channels and causes them to close. So potassium can now no longer go and diffuse in and out. So these potassium ion channels, we can call them ATP sensitive. So as the potassium ions can no longer diffuse out of the cell, the potential difference reduces, so depolarization occurs. Right, this depolarization causes calcium ion channels to open up, so now calcium ions can diffuse in. These calcium ions will go and bind to vesicles within the cell that contain insulin and cause these vesicles to go and move to the outer membrane of the cell and release the insulin by exocytosis.
Hopefully this video has given you some ideas how to teach this particular concept. If you've liked this video, please subscribe, like the video. There's links below to follow me on Facebook, Twitter, and there's a link below to my shop on test where you can go and get a full copy of this lesson. Please feel free to leave any comments or questions below and I will get back to you as quickly as possible. Thanks for watching and happy teaching.